Okay, hello everyone. Well, I'm going to go through a post that I recently made that I'm kind of excited about. Um, and I'll go through it and then hopefully we'll have some time for Q&A. Topic is what's special about life? Bulk orchestration and the Rulial Ensemble in biology and beyond. Towards a theory of bulk orchestration. It's a key feature of living systems, perhaps even in some ways the key feature, that even right down to a molecular scale, things are orchestrated. Molecules, or at least large ones, don't just move around randomly like in a liquid or a gel. Instead, what molecular biology has discovered is that there are endless active mechanisms that in effect orchestrate what even individual molecules in living systems do. But what is the result of all that orchestration? And could there perhaps be a general characterization of what happens in systems that exhibit such bulk orchestration? I've been wondering about these questions for some time, but finally now I think I may have the beginnings of some answers. The central idea is to consider the effect that being adapted for an overall purpose has on the underlying operation of a system. At the outset, one might imagine that there'd be no general answer to this, and that it would always depend on the specifics of the system and the purpose. But what we'll discover is that there is, in fact, typically a certain universality in what happens. Its ultimate origin is the principle of computational equivalence and certain universal features of the phenomenon of computational irreducibility that it implies. But the point is that so long as a purpose is somehow computationally simple, then more or less regardless of what in detail the purpose is, a system that achieves it will show certain features in its behavior. Whenever there's computational irreducibility, we can think of it as exerting a powerful force towards unpredictability and randomness, as it does, for example, in second law of thermodynamics. So, for example, for a system to achieve an overall computationally simple purpose, this computational irreducibility must in some sense be tamed, or at least contained. And in fact, it's an inevitable feature of computational irreducibility that within it there must be pockets of computational reducibility where simpler behavior occurs. And at some level, the way computationally simple purposes must be achieved is by tapping into those pockets of reducibility. When there's computational irreducibility, it means that there's no simple narrative one can expect to give of how a system behaves, and no overall mechanism one can expect to identify for it. But one can think of pockets of computational reducibility as corresponding to at least small-scale identifiable mechanisms. And what we'll discover is that when there's a simple overall purpose being achieved, these mechanisms tend to become more manifest. And this means that when a system is achieving an overall purpose, there's a trace of this even down in the detailed operation of the system. And that trace is what I've called mechanoidal behavior, behavior in which there are at least small-scale mechanism-like phenomena that we can think of as acting together through bulk orchestration to achieve a certain overall purpose. The concept that there might be universal features associated with the interplay between some kind of overall simplicity and underlying computational irreducibility is something not unfamiliar. Indeed, in various forms, it's the ultimate key to our recent progress in the foundations of physics, mathematics, and in fact, biology. In our effort to get a general understanding of bulk orchestration and the behavior of systems that achieve purposes, there's an analogy we can make to statistical mechanics. One might have imagined that to reach conclusions about, say, gases, we'd have to have detailed information about the motion of molecules. But in fact, we know that if we just consider the whole ensemble of possible configurations of molecules, then by taking statistical averages, we can deduce all sorts of properties of gases. And yes, the foundational reason this works, we can now understand in terms of computational irreducibility and so on. So could we perhaps do something similar for bulk orchestration? Is there some ensemble we can identify in which, wherever we look, there will be, with overwhelming probability, certain properties? In the statistical mechanics of gases, we imagine that the underlying laws of mechanics are fixed, but there's a whole ensemble of possible initial configurations for the molecules, almost all of which turn out to have the same limiting features. But in biology, for example, we can think of different genomes as defining different rules for the development and operation of organisms. And so now what we want is a new kind of ensemble that we can call a Rulial ensemble, an ensemble of possible rules. But in something like biology, it's not all possible rules we need. Rather, it's rules that have been selected to achieve the purpose of making a successful organism. We don't have a general way to characterize what defines biological fitness, but the key point here is that at a fundamental level, we don't need that. Instead, it seems that just knowing that our fitness function is somehow computationally simple tells us enough to be able to deduce properties of our Rulial ensemble. But are the fitness functions of biology in fact computationally simple? I've recently argued that their simplicity is precisely what makes biological evolution possible. Like so many other foundational phenomena, it seems that biological evolution is a reflection of the interplay between computational simplicity, in the case of fitness functions, this case of fitness functions, and underlying computational irreducibility. In effect, organisms have a chance only because the problems they have to solve aren't too computationally difficult. But now we can use the simplicity of fitness functions without knowing any more details about them to make conclusions about the relevant Rulial ensemble, and from there to begin to derive general principles associated with bulk orchestration. When one describes why a system does what it does, there are two different approaches one can take. One can go from the bottom up and describe the underlying rules by which the system operates, in effect, its mechanism, or one can go from the top down and describe what the system achieves, in effect, its goal or purpose. It tends to be difficult to mix these two approaches, but what we're going to find here is that by thinking in terms of the Rulial ensemble, we'll be able to see the general pattern of both the upward effect of underlying rules and the downward effect of overall purposes. What I'll do here is just a beginning, a first exploration, both computational and conceptual, of the Rulial ensemble and its consequences. But already there seem to be strong indications that by thinking in terms of the Rulial ensemble, one may be able to develop what amounts to a foundational theory of bulk orchestration, and with it, a foundational theory of certain aspects of adaptive systems, and most notably, biology. Okay, some first examples. Well, to begin our explorations and start developing our intuition, let's look at a few simple examples. We'll use the same basic framework that I did in my recent work on biological evolution. The idea is to have cellular automata whose rules serve as an idealization of genotypes and whose behavior serves as an idealization of the development of phenotypes. For our fitness function, we're going to start with something very specific, that after 50 steps, starting from a single cell seed, our cellular automaton should generate output that consists of 3 equal blocks of cells colored red, green, and yellow. Red, blue, and yellow. Okay, so let's show something here. So well, here are some examples of rules that solve this particular problem in the sense that in various different ways. So how can such rules be found? Well, we can use an idealization of biological evolution. So remember, the goal was to start from a single cell seed and generate a pattern that has a block of red, block of blue, block of yellow. So how can we find these rules? So we'll use an idealization of biological evolution. So let's start with the first rule that we had in the picture above. Let's understand how we get a picture like this. For example, we can start with a null rule and then make successive random point mutations in the rule, keeping the mutations that don't take us further from achieving our goal and dropping those that do. So each indicated here by red dots. So we're looking here, mutation number is along the x-axis, and we're showing with a red dot every candidate mutation, and we're keeping only those mutations that don't get us further away from our goal. So The result of that is that we progressively adapt over the course of a few thousand mutations here to successively reduce the number of errors and eventually, in this case perfectly, achieve our goal. So early in the sequence, there's lots of computational irreducibility in evidence. But in the process of adapting to achieve our goal, the computational irreducibility gets contained and in effect gets squeezed out until eventually the final solution in this case has an almost completely simple structure. So this example succeeds in exactly achieving the objective we defined, though it takes about 4,000 steps of adaptive evolution to do so. But if we limit the number of steps of adaptive evolution, then in general, we won't be able to reach the exact objective we've defined. So here are some results of what we get with 10,000 steps of adaptive evolution. It's kind of sorted by how close they get. So sometimes we make it perfectly, sometimes we don't. So in some of the cases here, there's a certain amount of kind of identifiable mechanism to what these rules do. There can be patches of complex and presumably computationally irreducible behavior, but particularly the rules that do better at achieving our exact objective tend to keep this computational irreducibility at bay and emphasize their simple mechanisms. So what we see is that among all possible rules, those that get even close to achieving the purpose we've set, in effect, show at least patches of mechanism. In other words, the imposition of a purpose selects out rules that exhibit mechanism and show what we've called mechanoidal behavior. The concept of mutational complexity. So we just saw that adaptive evolution can find, in this case, 4-color cellular automaton rules that generate this particular red, blue, yellow output we specified. What about other kinds of output? What we manage to get will depend on how much effort of adaptive evolution we put in. If we limit ourselves to 10,000 steps of adaptive evolution, here are some examples of what happens. So in these different cases, we're showing different objective functions and different extents to which in 10,000 steps of evolution, we're able to achieve those objective functions. The objective functions are the color blocks at the bottom of each row, and then we get to see how well we achieve those. So at a qualitative level, the main takeaway is that it seems that the simpler the objective is, like the ones at the top there, the more closely it's likely to be achieved by a given number of steps of adaptive evolution. So looking at the ultimately most successful examples here, that's the first column of examples, we can ask how these examples are reached in the course of adaptive evolution. I'm going to show more. This was more examples of what happens with more elaborate kinds of objective functions there, with 5 different color blocks at the top and then various kinds of periodic and more detailed color arrangements down below, showing showing the way in which the more complicated the objective, the more difficult it is to achieve in 10,000 steps. And those numbers say how many errors we have in relative to our objective in the final output here. Well, in any case, what we see, we can look now at how those sort of most successful results were achieved in the course of adaptive evolution. So we see that sort of the simpler sequences are reached both more successfully and more quickly. In effect, they seem to be easier for adaptive evolution to generate. But what do we mean by simpler in the context of sequences here? In qualitative terms, we can think of the simplicity of a sequence as being characterized by how short a description we can find of it. We might try to compress the sequence, say using some standard practical compression technique like run length encoding or block encoding or dictionary encoding. And for the sequences we're using here, these will mostly agree about what's simpler and what's not. And then what we find is that sequences that are simpler in this kind of characterization tend to be ones that are easier for adaptive evolution to produce. But actually, our study of adaptive evolution itself gives us a way to characterize the simplicity or complexity of a sequence. We can consider a sequence more complex if the typical number of mutations it takes to come up with a rule to generate the sequence is larger. And we can define this number of mutations to be what we can call the mutational complexity of a sequence. So there are lots of details in tightening up this definition, but in some sense what we're saying is that we can characterize the complexity of a sequence by how hard it is for adaptive evolution to get it generated. To get more quantitative, we have to address the issue that if we run adaptive evolution multiple times, it'll generally take different numbers of steps to be able to get to a particular sequence generated, or say to get to the point where there are fewer than m errors in the generated sequence. And sometimes, by the way, a particular run of evolution might get stuck and never be able to generate a particular sequence, at least, as we'll talk about later, with the kind of rules and single point mutations we're using so far. But we can still compute the probability across many runs of adaptive evolution to have reached a specified sequence within m errors after a certain number of steps. And what we can see here is how that probability builds up for the sequences, for these sequences that we were just looking at. So we can see here more quantitatively that some sequences are faster and easier to reach than others. We can go further by computing in each case the median number of adaptive steps needed to get within m errors of each sequence. So that's the result for that. That's the median number of steps required to get within a certain number of errors of these given sequences. So if we pick a certain desired fidelity, say allowing a maximum of 20 errors, then we can get at least one estimate of the mutational complexity for all our sequences. So we're plotting here kind of how difficult it is to get the x-axis is how difficult it is to get to a sequence, and then we're seeing a bunch of sequences here. Well, needless to say, these kinds of numerical measures are best a very coarse way to characterize the difficulty of being able to generate a given sequence through rules produced by adaptive evolution. And for example, instead of just looking at the probability to reach a given fidelity, we could be looking at all sorts of distributions and correlations. But in developing our intuition about the Rulial ensemble, it's useful to see how we can derive even an admittedly coarse, specific numerical measure of the difficulty of reaching a sequence through adaptive evolution. Well, let's talk about possible and impossible objectives. So we've seen that some sequences are easier to reach by adaptive evolution than others. But can any sequence we might look for actually be found at all? In other words, regardless of whether adaptive evolution can find it, is there in fact any cellular automaton rule at all, say a four-color one, that successfully generates any particular given sequence? Well, it's easy to see that in the end, there must be sequences that can't be generated with a cellular automaton. There are 4 to the 4 to the 3, in other words, 10 to the 38 possible four-color cellular automaton rules. But even though that's a large number, the number of possible four-color length 100 sequences is still much larger, like 4 to the 101, which is about 10 to the 61. So that means that it's inevitable that some of these sequences will not appear as the output from any four-color cellular automaton rule run from single-cell initial conditions for 50 steps. So we can think of such sequences as having too high an algorithmic complexity to be generated from a program as short as a four-color cellular automaton rule. But what about sequences that are simple with respect to our qualitative criteria above? Whenever we succeeded in what we talked about before in finding sequences by adaptive evolution, then we obviously know that they can be generated. But in general, this is a quintessentially computationally irreducible question. So that in effect, the only way to know for sure whether there's any rule that can generate a particular sequence is just to explicitly search through all the 10 to the 38 possible rules. We can get some intuition, though, by looking at a much simpler cases. So consider, for example, the 128 quiescent elementary cellular automata with two colors and range 1. So the number of distinct sequences that can be generated by those elementary cellular automata after T steps quickly stabilizes, and that's the result. That's the number of distinct sequences that can be generated by any of the quiescent cellular automata after a certain number of steps, and the maximum number of such sequences is just 32. But that number is soon much smaller than the total number of possible sequences of a given length, which is 2 to the 2t plus 1. So in this picture, the down wiggles are associated with additive rules like 90 and 150, which generate almost blank sequences at steps of the form 2 to the m. So let's see. Here's the corresponding result for two-color range 1.5 rules. That's 4 cells mapped to 1 cell type rules. So the question is, what are the sequences that get left out by cellular automata? Already by step 2, a quiescent elementary cellular automaton can only produce 14 of the 32 possible sequences, with sequences like white, black, and black, white being among the ones that are excluded. So one might think that one will be able to characterize excluded sequences just by saying that certain fixed blocks of cells could not occur at any step in a rule. And indeed, that happens for these R equals 3/2 rules. But for quiescent elementary cellular automata with R equals 1, range 1, it seems that if every block of any given any block of any given size will occur. Actually, no, I said that wrong. Back for the range 1/2 rules, that's just 2 cells mapping onto a single cell. There are cases where there's simply fixed blocks that can never occur. But as soon as you even get to the range 1 rules, that is rules where you're looking at a cell and its two neighbors, so a 3-cell neighborhood, it seems that every block of any given size will eventually occur, presumably as a result of the presence of rules like Rule 30 that generate large numbers of distinct blocks. But you can ask, for example, specifically about, let's say, periodic sequences. So you can say there are, and it turns out there are no quiescent elementary cellular automata that can generate sequences like these ones here. And yes, by most standards, these are quite simple sequences. They just happen to be not to be simple for elementary cellular automata. And indeed, we can expect that there will be plenty of such coincidentally unreachable but seemingly simple sequences, even for our 4-color rules. But we can also expect that even if we can't precisely reach some objective sequence, we'll still be able to get a sequence that's at least close. I mean, the minimum error is, for example, 4 cells out of 15 for the first sequence that we're looking at here, and 2 for the last sequence. So in other words, with elementary rules, we can get within 4 cells of that first sequence and within 2 cells of the last sequence that we're showing here. But there still remains the question of whether adaptive evolution will be able to find such sequences. For the very simple case of quiescent elementary cellular automata, we can kind of readily map out the complete multiway graph of all possible mutations between rules. And so this is what we get if we run all possible rules for 3 steps, then show possible outcomes as nodes here and possible mutations as edges. So the edges here are undirected because every mutation can go either way. That this graph has only 18 nodes reflects the fact that quiescent elementary cellular automata can only produce 18 of the 128 possible length 7 sequences. But even within these 18 sequences, there are ones that cannot be reached through the adaptive evolution process that we've been talking about. For example, let's say our goal is to generate a sequence, black, black, white, black, white, black, black, or rather to find a rule that can do that. If we start from the null rule, which generates just an all-white sequence, then our adaptive evolution process defines a foliated version of this multiway graph, a foliated version where you're always trying to get to something that is closer to your objective, or at least not further away from your objective. And then starting from that null rule at the top, the result of the null rule at the top, some paths, in other words, some sequences of mutations, successfully reach our objective. But this only happens about 25% of the time. The rest of the time, the adaptive evolution gets stuck at rules that give other final states. And the adaptive evolution can never reach the actual objective that we had. And we can kind of see that here, that things can get stuck and reach a state from which there is no further mutational evolution. Okay, so what happens if we look at larger cellular automaton rule spaces? Well, in those cases, we can't expect to trace the full multiway graph of possible mutations. And if we pick a sequence at random as our target, then for a long sequence, the overwhelming probability is that it won't be reachable at all by any cellular automaton with a rule of the given type. But if we start from a rule, say picked at random, and use its output as our target, then this guarantees that there's at least one rule that produces the sequence. And then we can ask how difficult it is for adaptive evolution to find a rule that works, most likely not the original one. So here are some examples with the original rule on the left and the best results found from 10,000 steps of adaptive evolution on the right. And what we see is fairly clear. When the pattern generated by the original rule looks simple, adaptive evolution can readily find rules that successfully produce the same output, so that the error that we're listing as a number there is small, albeit doing it sometimes in very different ways from the original rule that we'd used to find a sequence that was findable by a cellular automaton. But when the pattern generated by the original rule is more complicated, adaptive evolution typically won't be able to find a rule that exactly reproduces its output. And so, for example, in the cases that we're seeing here, many errors remain, even in the best results after 10,000 steps of adaptive evolution, in the case where the behavior is not what we might consider simple. So ultimately, this isn't terribly surprising. When we pick a cellular automaton rule at random, it'll often show computational irreducibility. In a sense, all we're seeing here is that adaptive evolution can't break computational irreducibility. Or put another way, computationally irreducible processes generate mutational complexity. Other objective functions. So far what we've been talking about is a particular type of goal, to have a cellular automaton produce a specified arrangement of cells after a certain number of steps. But what about other types of goals? Well, we'll look at several here. The general features of what will happen with them follow what we've already seen, but each of them will show some new effects and will provide some new perspectives. First, let's talk about vertical sequences. So as a first example, let's consider trying to match not the horizontal arrangement of cells, but vertical ones. In particular, the sequence of colors in the center column of the cellular automaton pattern. So here's an example of what we get by having a block of red cells followed by an equal block of blue cells. So the results here are quite diverse and creative, but it's notable that sort of in all cases here, there's a definite mechanism to be seen right around the center column, but there's all sorts of complexity away from the center column. But it's sufficiently contained that the center column itself can achieve its simple goal. And notice that in the case of all these rules, they achieve the goal of having the center column be a block of red followed by a block of blue. Well, things are similar if we ask to get 3 blocks of color rather than 2, though this goal turns out to be somewhat more difficult to achieve. It's also possible to get 4 blocks, like this, with rules again where they're simple right at the center column, but they can get more complicated elsewhere. Well, in general, the difficulty of getting a particular vertical sequence of blocks tends to track the difficulty of getting the corresponding horizontal sequence of blocks. Or in other words, the pattern of mutational complexity seems to be similar for sequences associated with horizontal and vertical goals. This also seems to be true for periodic sequences. I mean, if we alternating colors are fairly easy to achieve. There's a bunch of rules that achieve just straight cell by cell alternation of colors. There are many tricks that are possible to get that alternation of colors. It's kind of interesting to see how creative kind of adaptive evolution was at finding different kinds of nested patterns and stripes and patterns where just the edge of the pattern achieves it and so on. So if we have a sequence, let's say with period 5, it's pretty much the same story. When the period gets more comparable to the number of cellular automaton steps that we're sampling for, the solutions that we find tend to get wilder. And some are quite fragile and don't generalize beyond the original sequence of number of steps for which they were adapted. So all of these patterns here do successfully produce this center column as a periodic sequence. But if we, for 50 steps or whatever, but if we try and continue past that, some of them will successfully continue the periodic pattern and others will completely fail. They will not generalize. So this is a question of when we make this kind of adaptive evolution, to what extent does it generalize but beyond the specifics of what we trained it for? Or rather, what we adapted it to. All right, let's talk about another objective function, color frequencies and output. So the types of goals we've talked about so far have all involved trying to get exact matches to specified sequences. So what happens if we just ask for an average result? For example, what if we ask for all 4 colors to occur with equal frequency in our output, but allow them to be arranged in any way? With the same adaptive evolution setup as before, we rather quickly find solutions. And because we're running for 50 steps and getting patterns of it to 101, we're always off by at least 1 relative to the exact result of each 4 colors occurring for 25 cells. But so a few of these solutions seem to have figured out a mechanism to get all these colors equally, but others seem like they just happen to work. And indeed, taking the first 3 cases that we're showing here, here are the relative numbers of cells of different colors obtained at successive steps in the cellular automaton. So the pattern from before at the top there in the middle that looks consistently simple has equal numbers of colors numbers of each color at every step, but the others just happen to hit equality after running for exactly 50 steps, but on either side don't achieve equality. And actually, it turns out that all these solutions are in some sense quite fragile. I mean, change the color of just one cell, and one typically gets an expanding region of change that takes the output far from color equality. So how can we get rules that more robustly achieve our color equality objective? Well, one approach is to force the adaptive evolution to take account of possible perturbations by applying a few perturbations at every adaptive evolution step and keeping a particular mutation only if it neither it nor any of its perturbed versions has lower fitness than before. So here's an example of one particular sequence of rules that one gets in that way. And now if we apply perturbations to the final result, it doesn't change much. Well, it's notable that this sort of robust solution looks simple, and indeed that's common. With a few other examples, here are a few other examples of exact solutions where they've been forced to be robust. So in effect, it seems that requiring a robust exact solution forces out computational irreducibility, leaving only readily reducible patterns. If we relax the constraint of being an exact solution even a little, though, more complex behavior quickly creeps in. Let's talk about whole pattern color frequencies. So we just looked at trying to achieve particular frequencies of colors in the output of a cellular automaton after running for 50 steps. But what if we try to achieve certain frequencies of colors throughout the pattern produced by the cellular automaton? For example, let's say our goal is to have color frequencies in certain specific ratios, like 1 to 5 to 1 to 1. So adaptive evolution fairly easily finds good solutions for this. And in fact, it does so for any relative red level. So here are some other examples of relative red levels. But if we plot the median number of adaptive evolution steps needed to achieve these results, i.e. our approximation to mutational complexity, we see that there's a sort of systematic increase of mutational complexity with red level. In effect, the higher the red level, the more stringent the constraints we are trying to satisfy are, and the more steps of adaptive evolution it takes to do that. But looking at the actual patterns obtained at different red levels, we also see something else, that as the constraints get more stringent, the patterns seem to have computational irreducibility progressively squeezed out of them, leaving behavior that seems more and more mechanoidal. So, as another example along the same lines, consider some other goals here for certain ratios. So consider what happens if you vary the white level in these ratios of cell frequencies. So we see two rather different approaches being taken to the problem of having more white. When the white level isn't too large, the pattern just gets area with more white inside. But eventually the pattern tends to contract, leaving room for white outside. So it's kind of different ways that adaptive evolution solves the problem that we've set of increasing the white level. Let's talk about growth shapes. So what we've talked about so far, the overall shapes of our cellular automaton patterns have ended up always just being simple triangles that expand by one cell in each step, although we just saw that with sufficiently stringent constraints on colors, they're forced to be different shapes. The patterns we get are forced to be different shapes. But what if our actual goal is to achieve a certain shape? For example, let's say we try to get triangular patterns that grow at a particular rate on each side. So here are results for growth rates of the form 1 over n, i.e. growing by an average of one cell every n steps. So, and here's the sequence of adaptive evolution steps that led to that first example of growth rate 1/3. And so we see that we start off from rules that typically have growth rate 1, and we end up sort of grinding it down to eventually have growth rate 1/3. Here's the corresponding result for growth rate 1/10. And although the interiors of most of the final patterns that we had back here are complicated, their outer boundaries, well, yeah, even in this adaptive sequence where we're trying to reach sort of 1/10 angle here, even though the final patterns are pretty complicated inside, their outer boundaries tend to be simple, at least for when we have small, when we're not trying to make the thing too pointy. In a sense, what we're seeing back here is that we're sort of very mechanically, in some cases, generating the exact target growth rate. So we see that we see here that sort of the outer boundaries of these are really generating the growth rates, even the 1/7 growth rate, for example, in a very sort of mechanical way. Even though the interiors of the patterns may be more complicated, the outer boundaries have this definite kind of mechanism that's visible in them. So as we increase the value of n, we make things pointier, things do get more complicated, and adaptive evolution doesn't typically find an exact solution. So, and in fact, in the case there of 9 and 10, we don't have exact solutions where the outer boundary is repeatedly getting the result we wanted. And if we run those solutions longer, well, if we run the 1/2 solution longer and so on, it'll do just fine. if we run the 1/9 solution or the 1/10 solution longer, it doesn't do fine. Um, in other words, once again, we are with these particular constraints, we're not finding solutions that generalize well, and soon they start deviating from their target growth rates. So, in other words, as we increase n, we typically see that more steps of adaptive evolution are needed to achieve our goal. So the pointier things are it's reflecting the idea that larger n growth has more mutational complexity. So as a function of the pointiness, we're seeing the mutational complexity needed to achieve a satisfactory result. So for rational growth rates like 1/3, fairly simple exact solutions are possible. But for irrational growth rates, that's no longer true. Still, it turns out that adaptive evolution is in a sense strong enough, and our cellular automaton rule space is large enough that good approximations, even to irrational growth rates, can often be reached. So there are a few examples. Well, the solutions typically remain quite consistent when run longer, but there are still some notable limitations. For example, while a growth rate of exactly 1 is easy to achieve, growth rates close to 1 are in effect structurally difficult. For example, above about 0.741, adaptive evolution tends to cheat, kind of adding a little hat at the top of the pattern instead of actually producing a boundary with a consistent slope. Well, we talked about having sort of triangular shapes with certain opening angles as shapes. What about other shapes as goals? So here's what happens with diamond shapes of different widths. Adaptive evolution is quite constrained by what's structurally possible in a cellular automaton of the type we're talking about, and the results are not particularly good here. And indeed, if one attempts to generalize these results, it's clear none of them sort of really have the idea of generating a diamond shape. They all generalize to very different kinds of shapes. Lifetimes. So in what we've so far talked about, we focused on what cellular automata do over the course of a fixed number of steps, not worrying about what they might do later. But another goal we might have, which in fact I've talked about at length elsewhere, is just to have our cellular automata produce patterns that go for a certain number of steps and then die out. So for example, we can use adaptive evolution to find cellular automata whose patterns live for exactly 50 steps and then die out. Here are some examples. So just like in the other cases we talked about, adaptive evolution finds all sorts of diverse and interesting solutions to the problem of living for exactly 50 steps. Some, like the last one in this sequence and the yellow one, have a certain level of obvious mechanism to them. But most of these solutions seem to just work for no obvious reason. Presumably the constraint of living for 50 steps is in a sense just isn't stringent enough to squeeze out computational irreducibility, so there's still plenty of computational irreducibility in these results. What about mutational complexity? Approximating this, as we did before, by the median of the number of adaptive evolution steps and sampling a few hundred cases for each lifetime goal and plotting the quartiles as well as the median, we see sort of a systematic increase of mutational complexity as we increase the lifetime goal. So in effect, this shows us that if we increase the lifetime goal, it becomes more and more difficult for adaptive evolution to reach that goal. And as I've talked about elsewhere, if we go far enough, we'll eventually reach the edge of what's even in principle possible with, say, the particular type of four-color cellular automata we're using here. Well, all the objectives we've discussed so far have the feature that they're in a sense explicit and fixed. We define what we want, for example, a cellular automaton pattern that lives through exactly 50 steps, and then we use adaptive evolution to try to get it. But looking at something like lifetime suggests another possibility. Instead of our objective being fixed, our objective can instead be open-ended. And so, for example, we might ask not for a specific lifetime, but just to get the largest lifetime we can. So I talked about this at some length elsewhere, but how does it relate to the concept of the Rulial Ensemble that we're studying now? Well, when we have rules that are found by adaptive evolution with fixed constraints, we end up with something that can be thought of roughly analogous to things like the canonical, i.e. specified temperature ensemble of traditional statistical mechanics. But if instead we look at the winners of open-ended adaptive evolution, then what we have is more like a collection of extreme value extreme value points than something we can view as typical of the bulk of an ensemble. Periodicities. So we looked at the goal of having patterns that survive for a certain number of steps. Another goal we can consider is having patterns that periodically repeat after a certain number of steps. We can think of that as an extremely idealized analog of having multiple generations of a biological organism. Well, these are some examples of rules found by adaptive evolution that lead to patterns that repeat after exactly 50 steps. And as usual, there's a distribution in the number of steps of adaptive evolution required to achieve these results. This is the distribution. Those results they had were all exact results that repeat exactly after 50 steps. That's the distribution of the number of steps of adaptive evolution required to achieve those results. So looking at the median of the analogous distributions for different possible periods, we can get an estimate of the mutational complexity of different periods, which seems to increase somewhat uniformly with period. So it takes more and more steps of adaptive evolution to reach a particular period as the period gets larger. By the way, it's also possible to restrict our adaptive evolution so that it samples only symmetric rules. And so here are a few examples that kind of at least look nice of period 50 results that are found in that way. So in what we've talked about so far about periodicity, we've insisted on periodicity from the start, meaning that after each period, the pattern we get has to return to the single cell state we started from. But we can also consider periodicity that develops after a transient. Sometimes the transient is short, sometimes it's much longer. Sometimes the periodic pattern starts from a small seed, sometimes its seed is quite large. So here are examples of patterns found by adaptive evolution that have ultimate period 50. By the way, in all these cases, the periodic pattern seems like the main event of the cellular automaton evolution, but there are other cases where it seems more like a residue from other behavior. And indeed, that other behavior can in principle go on for arbitrarily long before finally giving way to periodicity. So we've been talking so far about the objective of finding cellular automaton rules that yield patterns with specific periodicity. But just like for lifetime, we can also consider the open-ended objective of finding rules with the longest periods we can. So here are the best results found with a few runs of 10,000 steps of adaptive evolution, where we're looking for periodicity without transients, and we're looking for the longest periodicity we can get. Okay, let's talk about measuring mechanoidal behavior. So we've now seen lots of examples of cellular automata found by adaptive evolution. And the key question is, what's special about them? If we look at cellular automata with rules purely chosen at random, well, here are some typical examples of what we'll see. Some of these patterns are simple, but many of them are complicated and in fact look quite random, though often with regions or patches of regularity. But what's striking is how visually different they look from what we've mostly seen in the cellular automata that we've looked at that were adaptively evolved for a purpose. So how can we characterize and ultimately measure that difference in appearance? Our randomly picked cellular automata seem to show either almost total computational reducibility and very trivial behavior, or unchecked computational irreducibility, albeit usually with regions or patches of computational reducibility. But cellular automata that were successfully evolved for a purpose tend to look different. They tend to show what we're calling mechanoidal behavior, behavior in which there are identifiable mechanism-like features, albeit usually mixed in with at least some, typically highly contained, sparks of computational irreducibility. At a visual level, there are typically some clear characteristics to mechanoidal behavior, For example, there are usually repeated motifs that appear throughout a system. And there's also usually a certain degree of modularity, with different parts of the system operating at least somewhat independently. And yes, there's no doubt a rich phenomenology of mechanoidal behavior to be studied, closely related to the study of class 4 behavior that I started looking at in the 1980s. But at a coarse and potentially more immediately quantitative level, a key feature of mechanoidal behavior is that it involves a certain amount of regularity and computational reducibility. So how can one measure that? Whenever there's regularity in a system, it means there's a way to summarize what the system does more succinctly than just by specifying every state of every element of the system. So in other words, there's a way to compress our description of the system. Well, perhaps one might think one could use modularity to do this compression, say by keeping only the modular parts of a system and eliding away the filler in between, much like run length encoding. But like run length encoding, the most obvious version of this runs into trouble when the filler consists, for example, of alternating colors of cells. One can also think of using block-based encoding or dictionary encoding, say leveraging repeated motifs that appear. But it's an inevitable feature of computational irreducibility that in the end, there can never be one universally best method of compression. But as a specific example, let's use the Wolfram language compress function. If you use gzip or bzip2 and so on, we get essentially identical results. So feed in a cellular automaton pattern, and the compress function will give us a losslessly compressed version of it. And then we can use the length of this compressed version as a measure of what's left over after we compress out the regularities in the pattern. So here's a plot of the compressed description length in compress output bytes for some of the patterns we've talked about before here. And what's immediately striking is that the patterns that were sort of evolved for a purpose tend to be in between the patterns that come from randomly chosen rules. And yes, the question of what's compressed by what is a complicated, somewhat circular story. Compression is ultimately about making a model for things that one wants to compress. And for example, the appropriate model will change depending on what those things are. But for our purposes here, we'll just use the compress function, which is set up to do well at compressing sort of typical human relevant content. Okay, so how does adaptive evolution relate to our compressed size measure? So here's an example of a typical progression of an adaptive evolution process, in this case, based on the goal of generating our first red, blue, yellow sequence. So given our way of starting with a null rule, everything is simple at the beginning, yielding a small compressed size. But soon the system starts developing computational irreducibility, and the compressed size goes up. Still, as the adaptive evolution proceeds, the computational irreducibility is progressively squeezed out, and the compressed size settles down to a smaller value. So this plot is based on a particular randomly chosen sequence of mutations in the underlying rule. But if we look at the average from a large collection of mutation sequences, shown here, we see much the same kind of thing. Even though the error rate on average goes down monotonically, the compressed size of our candidate patterns has a definite peak before settling down to its final value. In effect, it seems that the system needs to explore the computational universe a bit before figuring out how to achieve its goal. How general is this? If we don't insist on reaching zero error, we get a curve of the same general shape, slightly smoothed out here because we're allowing 20 or fewer errors. What about for our other target sequences? So here are results for all the target sequences we talked about before, with the number of errors allowed in each case indicated. In all these cases, we get final compressed sizes that are much smaller than those for the all but very simple randomly chosen rules, indicating that our adaptive evolution process has indeed generated regularity, and our compression method has successfully picked this up. Well, looking at the overall shape of the curves we've generated here, there seems to be a general phenomenon in evidence. The process of adaptive evolution seems to pass through a computationally irreducible period before getting to its final mechanoidal phase. Even as it gets progressively closer to its goal, the adaptive evolution process ends up still playing the field before homing in on its final solution. And yes, phenomena like this are seen in both the fossil record of life on Earth and in the development of engineering systems. Well, let's talk about the Rulial Ensemble and its implications. So we began by asking the question, what consequences does being evolved for a purpose have on a system? So we've now talked about many examples of different purposes and seen many and seen sort of how adaptive evolution can achieve those purposes. So the details are different in different cases, but we've seen that there are general features that occur very broadly. Perhaps the most notable feature is that in systems that were adaptively evolved for a purpose, one tends to see what we can call mechanoidal behavior, behavior in which there are mechanism-like elements. The rules that are picked purely at random, in effect uniformly from Rulial space, rarely show mechanoidal behavior. But in rules that have been adaptively evolved for a purpose, mechanoidal behavior is the norm. And what we found here is that this is true essentially regardless of what the specific purpose involved is. We can think of rules that have been adaptively evolved for a purpose as forming what we can call a Rulial ensemble, a subset of the space of all possible rules, concentrated by the process of adaptive evolution. And what we've seen is that there are, in effect, generic features of the Rulial ensemble, features that are generically seen in rules that have been adaptively evolved for a purpose. Given a particular purpose, like produce this specific sequence as output, there are typically many ways it can be achieved. It could be that one can engineer a solution that shows a human recognizable mechanism all the way through. It could be that it will be a lump of computationally irreducible behavior that somehow just happens to have the result of doing what we want. But adaptive evolution seems to produce solutions with what amount to intermediate characteristics. There are elements of mechanism to be seen, and there are also usually certain sparks of computational irreducibility. Typically, what we see is that in the process of adaptive evolution, all sorts of computational irreducibility is generated, but to achieve whatever purpose has been defined requires taming that irreducibility. And it seems introducing the kind of patches of mechanism that are characteristic of the mechanoidal behavior we've seen. That those patches of mechanism fit together to achieve an overall purpose is often a surprising thing, but it's the essence of the kind of bulk orchestration that we see in systems we've studied, and that also seems to be characteristic of biological systems. So having specified a particular purpose, it's often completely unclear how a given kind of system could possibly achieve it. And indeed, what we've seen adaptive evolution do here often seems like magic. But in a sense, what we're seeing is just a reflection of the immense power that is available in the computational universe and manifest in the phenomenon of computational irreducibility. Without computational irreducibility, we'd somehow get what we expect. Computational irreducibility is what adds what is ultimately an infinite element of surprise. And what we've seen is that adaptive evolution manages to successfully harness that element of surprise to achieve particular purposes. So let's say our goal is to generate a particular sequence of values. One might imagine that just operating on a space of possible sequences and gradually adaptively evolving to the sequence we want. But that's not the setup we're using here, and it's not the setup biology has either. Instead, what's happening both in our idealized cellular automaton systems and in biology is that the adaptive evolution process is operating at the level of underlying rules, but the purposes are achieved by the results of running those rules. And it's because of this distinction, which in biology is associated with genotypes versus phenotypes, that computational irreducibility has a way to insert itself. In some sense, both the underlying rules and overall patterns of purposes can be thought of as defining certain syntactic structures. The actual running of the rules then represents what one can think of as the semantics of the system, and it's there that the power of computation is injected. But in the end, just how powerful is adaptive evolution with its ability to tap into computational irreducibility and to mine the computational universe. We've seen that some goals are easier to reach than others. And indeed, we introduced the concept of mutational complexity to characterize just how much effort of adaptive evolution and ultimately how many mutations is needed to achieve a given objective. If adaptive evolution is able to fully run its course, then we can expect it to achieve its objective through rules that show mechanoidal behavior and clear evidence of mechanism. But if the amount of adaptive evolution stops short of what's defined by the mutational complexity of the objective, then one's likely to see more of the untamed computational irreducibility that's characteristic of intermediate stages of adaptive evolution. In other words, if we get all the way to a solution, there'll be mechanism to be seen. But if we stop short, there's likely to be all sorts of gratuitous complexity associated with computational irreducibility. We can think of our final Rulial ensemble as consisting of rules that successfully achieve their purpose. When we stop short, we end up with another intermediate Rulial ensemble consisting of rules that are merely on a path to achieve their purpose. In the final Rulial ensemble, the forces of adaptive evolution have in a sense tamed the forces of computational irreducibility. But in the intermediate ensemble, the forces of computational irreducibility are still strong, bringing the various universal features of computational irreducibility to the fore. It might be useful to contrast all of this with what happens in traditional statistical mechanics, say molecules in a gas, which I've argued elsewhere, in which I've argued that there's almost always unchecked computational irreducibility. And it's this unchecked computational irreducibility that leads to the second law by producing configurations of the system that we, as computationally bounded observers, can't distinguish from random and can therefore reasonably model statistically just as being typical of the ensemble, where now the ensemble consists of all possible configurations of the system that, for example, have the same energy or the same temperature. Adaptive evolution is a different story. First, it's operating not directly on the configurations of a system, but rather on the underlying rules for the system. And second, if one takes the process of adaptive evolution far enough, so that, for example, the number of steps of adaptive evolution is large compared to the mutational complexity of the goal one's pursuing, then adaptive evolution will tame the computational irreducibility. But there's still an ensemble involved, though now it's an ensemble of rules rather than an ensemble of configurations. And it's not an ensemble that somehow covers all rules, rather it's an ensemble that's sculpted by the constraint of achieving a purpose. So I've argued that it's the characteristics of observers like us that ultimately lead to the perceived validity of the second law. So is there a role for the notion of an observer in what we've been discussing here, a bulk orchestration in the Rulial ensemble? Well, yes. And in particular, it's critical in grounding our concept of purpose. We might ask, what possible purposes are there? Well, for something to be a reasonable purpose, there has to be some way to decide whether or not it's been achieved. And to make such a decision, we need something that's in effect an observer. In the case of statistical mechanics and the second law, and in fact in all our recent successes in deriving foundational principles in both physics and mathematics, we want to consider that observers that are somehow like us, because in the end, what matters for us is how we as observers perceive things. So I've argued that the most critical feature of observers like us is that we're computationally bounded, and also somewhat relatedly, that we assume we're persistent in time. And it then turns out that the interplay of these features with underlying computational irreducibility is what seems to lead to the core principles of physics and mathematics that we're familiar with. But what should we assume about the observer or the determiner of purpose in adaptive evolution and in particular in biology? It turns out that once again, it seems as if computational boundedness is the key. In biological evolution, the implicit goal is to have a successful or fit organism that will, for example, reproduce well in its environment. But the point is that this is a coarse constraint that we can think of at a computational level as being computationally bounded. And indeed, I've recently argued elsewhere that it's this computational boundedness that's ultimately responsible for the fact that biological evolution can work at all. If to be successful, an organism always immediately had to satisfy some computationally very complex constraint, adaptive evolution wouldn't typically ever be able to find the necessary solution. Or put another way, biological evolution works because the objectives it ends up having to achieve are of limited mutational complexity. But why should it be that the environment in which biological evolution occurs can be navigated by satisfying computationally bounded constraints? In the end, it's a consequence of the inevitable presence of pockets of computational reducibility in any ultimately computationally irreducible system. Whatever the underlying structure of things, there will always be pockets of computational reducibility to be found, and it seems that that's what biology and biological evolution rely on. Specific types of biological organisms are often thought of as populating particular niches in the environment defined by our planet. What we're saying here is that all possible evolved entities populate an abstract meta-niche associated with possible pockets of computational reducibility. But actually, there's more, because the presence of pockets of computational reducibility is also what ultimately makes it possible for there to be observers like us at all. As I've talked about elsewhere, it's an abstract necessity that there must exist a unique object that I call the Ruliad that is the entangled limit of all possible computational processes. And everything that exists must somehow be within the Ruliad. But where then are we? It's not immediately obvious that observers like us, with a coherent existence, would be possible within the Ruliad. But if we are to exist there, we must in effect exist in some computationally reducible slice of the Ruliad. And yes, for us to be the way we are, we must in effect be in such a slice. But we can still ask why and how we got there. And that's something that's potentially informed by the notion of adaptive evolution that we've discussed here. Indeed, what we've argued that for adaptive evolution to be successful, its objectives must in effect be computationally reducible. So as soon as we know that adaptive evolution is operating, it becomes in a sense inevitable that it will lead to pockets of computational reducibility. That doesn't in and of itself explain why adaptive evolution happens, but in effect it shows that if it does, it will lead to observers that at some level have characteristics like us. So then it's a matter of abstract scientific investigation to show, as in effect we've done here, that within the Ruliad it's at least possible to have adaptive evolution. Let's talk about purpose versus mechanism and the nature of life. So any phenomenon can potentially be explained both in terms of purpose and in terms of mechanism. Why does a projectile follow that trajectory? One can explain it as following the mechanism defined by its laws of motion. Or one can explain it as following the purpose of satisfying some overall variational principle, say extremizing the action associated with the trajectory. Sometimes phenomena are more conveniently explained in terms of mechanism, sometimes in terms of purpose. And one can imagine that the choice could be determined by which is somehow the computationally simpler explanation. But what about the systems and processes we talked about today? If we just run a system like a cellular automaton, we always know its mechanism. It's just its underlying rules. But we don't immediately know its purpose. And indeed, if we pick an arbitrary rule, there's no reason to think that it will have a computationally simple purpose. In fact, insofar as the running of the cellular automaton is a computationally irreducible process, we can expect that it won't achieve any such computationally simple purpose that we can identify. But what if the rule is determined by some process of adaptive evolution? Well, then the objective of that adaptive evolution can be seen as defining a purpose that we can use to describe at least certain aspects of the behavior of the system. But what exactly are the implications of the presence of purpose on how the system operates? The key point that's emerged here is that when there's a computationally simple purpose that's been achieved through a process of adaptive evolution, then the rules for the system will be part of what we've called the Rulial Ensemble. And then what we've argued is that there are generic features of the Rulial Ensemble, features that don't depend on what specific purpose the adaptive evolution might have achieved, only that its purpose was computationally simple. And foremost among these generic features is the presence of mechanoidal behavior. In other words, so long as there is an overall computationally simple purpose, what we found is that whatever in detail that purpose might have been, the presence of the purpose tends to push itself down to produce behavior that locally is mechanoidal in the sense that it shows evidence of visible mechanism. What do we mean by visible mechanism? Operationally, it tends to be mechanism that's readily amenable to human-level narrative explanation. It's worth remembering that at the lowest level, the system we've studied is set up to have simple mechanisms in the sense that they have simple underlying rules. But once these rules run, they generically produce computationally irreducible behavior that doesn't have a simple narrative-like description. But when we're looking at the results of adaptive evolution, we're dealing with a subset of rules that are part of the Rulial ensemble, and so we end up seeing mechanoidal behavior with at least local narrative descriptions. So as I've talked about, though, if the adaptive evolution hasn't entirely run its course, in the sense that the mutational complexity of the objective is higher than the actual amount of adaptive evolution that's been done, then there'll still be computational irreducibility that hasn't been squeezed out. So how does all this relate to biology? The first key element of biology, as far as we're concerned here, is that there's a separate genotype and phenotype related by what's presumably, in general, the computationally irreducible process of biological growth and so on. The second key element of biology for our purposes here is the phenomenon of self-reproduction, in which new organisms are produced with genotypes that are identical up to small mutations. Both these elements are immediately captured by the simple cellular automaton model we've used, and given them, we seem to be led inexorably to our conclusions here about the Rulial ensemble, mechanoidal behavior, and bulk orchestration. It's often been seen as mysterious how there ends up being so much apparent complexity in biology. But once one knows about computational irreducibility, one realizes that actually complexity is quite ubiquitous. And instead, what's in many respects more surprising is the presence of any explainable mechanism. But what we've seen here through the Rulial ensemble is that such explainable mechanism is in effect a shadow of the overall simple computational purposes. At some level, there's computational irreducibility everywhere, and indeed it's the driver for rich behavior. But what happens is that in the presence of overall purpose, patches of computational irreducibility have to be fitted together to achieve that purpose. Or in other words, there's inevitably a certain bulk orchestration of all those patches of computation. And that's what we see so often in actual biological systems. And so what is it in the end that's special about life and biological systems? I think more than anything, it's that it's involved so much adaptive evolution. All those 10 to the 40th individual organisms in the history of life on Earth have been links in chains of adaptive evolution. It might have been that all that effort of adaptive evolution would have the effect of just solving the problem and producing some simple mechanistic solution. But it's not what we've seen here, and it's not how we can expect things to work. Instead, what we have is an elaborate interplay of lumps of computational irreducibility being harnessed by simple mechanisms. It matters that there's been so much adaptive evolution, and what we are now seeing throughout biology, down to the smallest features, is the consequences of that adaptive evolution. Yes, there are frozen accidents of history to be seen, but the point is not those individual items, but rather the whole aggregate consequence of adaptive evolution, mechanoidal behavior, and bulk orchestration. And it's because these features are generic that we can hope they can form the basis for what amounts to a robust bulk theory of biological systems and biological behavior, something that has roughly the same bulk theory character as the gas laws or fluid dynamics. But that's now talking about systems that have been subject to large-scale adaptive evolution at the level of their rules. So what I've talked about here is very much just a beginning, but I think that the ruliological investigations that I've shown and the general framework I've described provide kind of the raw material for something we've never had before, a well-defined and general sort of fundamental theory for the operation of living systems. It won't describe all the historical accident details, but I'm hopeful it will provide a useful global view of what's going on that can potentially be harvested for answers to all sorts of useful questions about the remarkable phenomenon that we call life. Well, in what I wrote, a couple more sections. One is an appendix about different adaptive strategies. So let me show you a little bit about that. So, in our explorations of Rulial Ensemble and of mutational complexity, we've looked at a range of possible objective functions, but we've always considered just one adaptive evolution strategy, accepting or rejecting the results of single-point mutations in the rule at each step. So what would happen if we were to adopt a different strategy? The main conclusion is, it doesn't seem to matter much. For a particular objective function, there are adaptive evolution strategies that get to a given result in fewer adaptive steps, or that ultimately get further than our usual strategy ever does. But in the end, the basic story of the Rulial ensemble and of mutational complexity is robust relative to detailed changes in our adaptive evolution process. So, as an example, let's consider allowing more than one random mutation in the rule at each adaptive evolution step. Let's say, as when we at the beginning of what we were talking about, that our objective is to generate a red, blue, yellow output after 50 steps. So with just one mutation at each step, it's only a small fraction of adaptive evolution runs that we make that reach perfect solutions. But with more mutations at each step, the more progress is made, at least for this particular problem. And we can see here in these sort of histograms the number of errors remaining after 10,000 evolution steps. By the time we're dealing with by the time we're dealing with 5 mutations per step, we get the probability, the number of errors is peaked around 0 errors, whereas for 1 mutation per step, the number of errors is peaked around 60, although there's a tail that goes down to having gotten a perfect answer. Well, looked at in terms of multiway systems, having fewer mutations at each step leads to fewer paths between possible rules and a greater possibility of getting stuck. With more mutations at each step, there are more paths overall and a lower possibility of getting stuck. What about mutational complexity? If we still say that each adaptive evolution step accounts for a single unit of mutational complexity, even if the actual step involves multiple underlying mutations in the rule, here's what we get for the mutational complexity for different objectives. Here's how that's affected by having different numbers of overall mutations at each step. So the height of each tick here indicates the number of mutations that we're allowing. And this is showing the mutational complexity deduced by different adaptive evolution strategies for these different objectives. So yes, different numbers of mutations at each step lead to mutational complexities that are different in detail, but in most cases, surprisingly similar overall. They're surprising clustering of the results for different numbers of mutations per step. Well, what about completely different adaptive evolution strategies? There are many one could consider. For example, a gradient descent approach, where at each step we examine all possible mutations and pick the best one, i.e. the one that increases fitness the most. We can extend this by keeping not just the top rule at each step, but say the top 5 in an analog of beam search. There's also kind of a collaborative approach where multiple different paths of random mutations are allowed, but where every so often all are reset to be the best found so far. And indeed, we can consider all sorts of techniques from reinforcement learning, for example. In particular cases, any of these approaches can have significant effects. But in general, the phenomenon we're discussing here seem robust enough that the details of how adaptive evolution is done don't matter much. And the single mutation strategy we mostly used can be considered adequately representative. Well, I had a final section here about historical and personal background. It's been obvious since antiquity that living organisms contain some kind of gooey stuff that's different from what one finds elsewhere in nature. But what is it, and how universal might it be? In Victorian times, it had various names, the most notable being protoplasm. But increasing effectiveness of microscopy made it clear that there was actually a lot of structure in living matter, as, for example, reflected in the presence of different organelles. But until the later part of the 20th century, there was a general belief that fundamentally what was going on in life was chemistry, or at least biochemistry, in which all sorts of different kinds of molecules were randomly moving and undergoing chemical reactions at certain rates. The digital nature of DNA, discovered in 1953, slowly began to erode this picture, adding in ideas about molecular scale mechanisms and machinery that could be described in mechanical or informational rather than bulk statistical terms. And indeed, a notable trend in molecular biology over the past several decades has been the discovery of more and more ways in which molecular processes and living systems are actively orchestrated and not just the result of random statistical behavior. When a single component can be identified, cell membranes, microtubules, biomolecular condensates, and so on, there's quite often been bulk theory developed, typically based on ideas from statistical mechanics. But when it comes to constellations of different kinds of components, most of what's been done has been to collect the material to fill many volumes of biology texts with what amounts to narrative descriptions of how in detail things operate, with no attempt at any kind of general picture independent of particular details. Well, my own foundational interest in biology goes back about half a century. When I first started studying cellular automata at the beginning of the 1980s, I certainly wondered, as others had before, whether they might be relevant to biology. That question was then greatly accelerated when I discovered that even with simple rules, cellular automata could generate immensely complex behavior, which often looked visually surprisingly organic. In the 1990s, I put quite some effort into what amounts to macroscopic questions in biology. How do things grow in different shapes, produce different pigmentation patterns, and so on? But somehow in everything I studied, there was a certain assumed uniformity. Many elements might be involved, but they were all somehow essentially the same. Well, I was well aware of the complex reaction networks and later pieces of molecular machinery that had been discovered, but they felt more like systems from engineering, with lots of different detailed components, and not like systems where there could be a broad theory. Still, within the type of engineering I knew best, namely software engineering, I kept wondering whether there might perhaps be general things one could say, particularly about the overall structure and operation of large software systems. I made measurements, I constructed dependency graphs, I thought about analogies between bugs and diseases, but beyond a few power laws and the like, I never really found anything. A lot changed, however, with the advent of our physics project in 2020, because among other things, there was now the idea that everything, even the structure of space-time, was dynamic and in effect emerged from a graph of causal relationships between events. So what about biology? Could it be that what mattered in molecular biology was the causal graph of interactions between individual molecules? Perhaps there needed to be a subchemistry that tracked specific molecules rather than just molecular species. And perhaps to imagine that ordinary chemistry could be the basis for biology was as wide of the mark as thinking that studying the physics of electron gases would let one understand microprocessors. Back in the early 1980s, I had identified that the typical behavior of systems like cellular automata could be divided into four classes, with the fourth class having the highest level of obvious complexity. And yes, the visual patterns produced in class 4 systems often had a very lifelike appearance. But was there a foundational connection? In 2023, as part of closing off kind of a 50-year personal journey, I'd been studying the second law of thermodynamics and identified class 4 cellular automata as ones that, like living systems, aren't usefully described just in terms of a tendency to randomization. Reversible class 4 systems made it particularly clear that in class 4 there really was a distinct form of behavior that I called mechanoidal, in which a certain amount of definite structure and mechanism are visible, albeit embedded in great overall complexity. At first, from thinking about molecules, I made some surprise progress in 2024 on biological evolution. In the mid-1980s, I'd considered modeling evolution in terms of successive small mutations to things like cellular automaton rules. But at the time, it didn't work. And it's only now, after new intuition from the success of machine learning, that in 2024, I tried again, and this time it worked. Given an objective like make a finite pattern that lives as long as possible, adaptive evolution by successive mutation of rules say in a cellular automaton, seem to almost magically find typically very elaborate solutions. Soon I'd understood that what I was seeing was essentially raw computational irreducibility that just happened to fit the fairly coarse fitness criteria I was using. And I then argued that the success of biological evolution was the result of the interplay between computationally bounded fitness and underlying computational irreducibility. But okay, what did that mean for the innards of biological systems? Exploring an idealized version of medicine on the basis of my minimal models of biological evolution led me to look in a bit more detail at the spectrum of behavior in adaptively evolved systems and invariants produced by perturbations. But there was something general, something potentially universal. Was there something general, something potentially universal that one could say? In the second law, one often talks about how behavior one sees is somehow overwhelmingly likely to be typical of the ensemble of all possibilities. In the usual second law, the possibilities are just different initial configurations of molecules and so on. But in thinking about a system that can change its rules, the ensemble of all possibilities is, at least at the outset, much bigger. In effect, it's the whole Ruliad. But the crucial point I realized a few months ago is that changes in rules that are relevant for biology must be ones that can somehow be achieved by a process of adaptive evolution. But what should the objective of that adaptive evolution be? One of the key lessons from our physics project and the many things informed by it is that yes, the character of the observer matters, but knowing just a little about an observer can be sufficient to deduce a lot about the laws of behavior that observer will perceive. So that led to the idea that this kind of thing might be true about objective functions, and that just knowing that, for example, an objective function is computationally simple might be enough to tell one things about the Rulial ensemble of possible rules. But is that really true? Well, as I've done so many times, I turn to computer experiments to find out. In my work on the foundations of biological evolution, I'd used what are in a sense very generic fitness functions, like overall lifetime. But now I needed a whole spectrum of possible fitness functions, some of them by many measures, in fact, even simpler to set up than something like lifetime. So the results I've talked about today, I consider very encouraging. The underlying details don't seem to matter much, but a broad range of computationally simple fitness functions seem to worm their way down to have the same kind of effect on small-scale behavior and to make it, in a sense, seem mechanoidal. Biology has tended to be a field in which one doesn't expect much in the way of formal theoretical underpinning. And indeed, even after all the data that's been collected, there are remarkably few big theories in biology. Natural selection and the digital informational nature of DNA have been pretty much the only examples. But now, from the type of thinking introduced by a physics project, we have something new and different. The concept of the Rulial ensemble and the idea that essentially just from the fact of biological evolution, we can talk about certain features of how adaptively evolved systems in biology must work. Traditional mathematical methods have never gotten very far with the broad foundations of biology, and nor in the end has specific computational modeling. But by going to a higher level and in a sense thinking about the space of all possible computations, I believe we can begin to see a path to a powerful general framework, not only for the foundations of biology, but for any system that shows bulk orchestration produced by a process of adaptive evolution. And that's all I wrote. So I'm happy to try and address some questions and comments. I will say, as I read through this this piece of writing, I wrote it very quickly. A lot of help, I should acknowledge, from Willem Nielsen, who's been, I think, on this live stream interacting with folks. This project was eventually done in just a couple of weeks. And as I read through it, I realize it has a lot of depth and also has a lot of height, in a sense that it involves a lot of concepts that I've been sort of building in this big tower for a very long time. I think this is fairly important stuff, and I think it really has a lot that it can say about kind of where one can go with biology. But as I recognize in going through it, it is quite conceptually complex, and so it has many different twists and turns and so on. I hope people were able to follow them. All right. on Alexander. Alexander asks, if mechanoidal behavior is a generic signature of systems evolved under computationally simple objectives, which biological domain do you expect to show this most clearly? Gene regulatory interaction rules, protein assembly mechanisms, developmental patterning. Where would we expect to see the greatest difference in compressed description length between evolved rule systems and randomly parametrized ones? That's a great question. I don't have a great answer. It's something we are actively looking at, is kind of making connections between this sort of foundational model and actual issues in biology. I I would say um, when one is trying to understand kind of complexity, it's really useful to be able to kind of uh, experience it. And when one is doing things where all one can get from uh, experimental observations is either numerical data, which doesn't have the richness to sort of see complexity, or things like network graphs, which we humans have a very hard time kind of disentangling. The things that we always have the easiest time disentangling are things that kind of match with our visual system, two-dimensional pictures, things like this. So in places in biology where we can take the phenomena we see, the things we measure with all kinds of different measuring modalities, and map them in a fairly faithful way onto kind of two-dimensional images that we can kind of experience with our eyes, I think those are the most likely places to make quick progress. And I think that often will be developmental kinds of things and actual morphology of organisms, more so than reaction networks, which will be this thing that may have exactly this kind of behavior, but it's kind of hard to visualize. It's hard to tell what's going on in such a system. And I think in terms of protein assemblies and so on, that is again a bit more visual, but it's also rather three-dimensional. And also, one always has to remember with proteins that even though one sees pictures, this is the shape of a protein. Well, that was the shape of a protein when it was crystallized or when it was frozen in cryo-EM. But an actual protein in the wild is always flopping around in quite complicated ways. And as one starts thinking about sort of that video, that movie, as well as thinking about the three-dimensionality, I think it may become difficult to tell what's going on. So I kind of think, the thing you can get on the two-dimensional microscope slide is a good place to start because it lets one have this kind of experiential connection to the complexity of the behavior. Let's see. Brady asks, in our universe, is the initial purpose replication because it is the one that inevitably snowballs on itself? That is an interesting question. In other words, given self-replication, I've argued that it is somewhat inevitable that you end up with observers a bit like us. So then you can ask, what other principles might lead to very different kinds of observers? Is replication, is the idea of making copies of things that may be the seed idea that leads to observers like us? And that idea has more legs than one might imagine. I mean, for example, the concept that there is an objective reality in the world. If there was only one of us, one mind in a vat somewhere, it would be very unclear what it would even mean to talk about objective reality in the world. It's because there are many of us who are pretty similar that we can all sort of have an objective reality that we can agree about. So, and that is the fact that there are many of us that are kind of similar is a consequence of the phenomenon of self-reproduction. So I think it's a really interesting question, really interesting kind of philosophical question, whether whether there is a different basis that would lead to a very different experience. It might be an experience in which there is no objective reality, which, that objective reality might be a consequence of the fact that there are many subjective realities which and we're looking at sort of the intersection of those subjective realities. That's an interesting philosophical question. I have to think more about that. Ontological asks, what is the purpose of life and intelligence in the whole project of existence? Well, existence is a funky thing. My belief is that the Ruliad is an abstract construct that inevitably exists, in the same sense that, a 2 plus 2 equals 4, given that you've defined the notion of 2 and plus and so on, it's inevitable that 2 plus 2 equals 4. So too, it's inevitable that given you've defined the notion of computation, that the Ruliad exists. So the existence of the Ruliad is not something contingent, it's something necessary, it's something inevitable. The question of whether there exist observers like us in the Ruliad, that is a seemingly contingent fact. It could be the case that there wouldn't be observers like us. But what I've begun to argue here is that that too is a necessary fact, that given the Ruliad with all its richness, it is inevitably the case that there will be phenomena like self-reproduction, which will inevitably lead to observers like us. And that therefore, in a sense, our nature and the way that we observe the universe and the laws of physics we observe are necessary. They're not the only ones you could have. There could be that we, from our inner experience, we are part of this collection of entities that have been created by this process of self-reproduction and so on. And from our inner experience, that's the little zone of the Ruliad that we get to sample and whose properties we get to see. But there are many other zones of the Ruliad that work completely differently. They're not what is connected to our inner experience, but they're what's out there that could be connected to other experiences. Now, can there be other experiences? Or does even the notion of experience require a kind of coherent existence that relies on phenomena ultimately like self-reproduction? I think I've argued elsewhere that a critical feature of sort of our experience of consciousness, our coherent experience, the experience of having experience, that is a consequence of the fact that we're taking all this input data about the world and our brains, our minds are concentrating that down to this small thread of experience that leads us to do the things we do, to take the actions we take. We're taking all this input and using that to decide what our next action will be. And so I think this question of sort of that phenomenon of going from all of the outside to this thread of experience inside, I think that is essential to our notion of coherent existence for ourselves. In other words, it's much less clear that an organism, even maybe a biological one, that has lots of different cells doing different things, but doesn't have sort of a coherent central decision about what's going on. Us animals that move around have the issue of, are we going to move left or right? Are we going to take all those trillions of cells that are in us and walk left or walk right? If those cells were just sort of spread out in some biofilm somewhere, it wouldn't be the case that we have to have that collective decision about what we do. Each cell could be for itself. But for us, as organisms that can collectively, that where all our cells move around together, we have to make that decision coherently. And I think that's sort of the essence of what leads to this kind of notion of what we experience as this thread of experience, this single thread of experience, this coherent existence that we feel about ourselves. So that seems to be a necessary piece of the picture of what it takes to have an experience like ours. Now, as we go away from that idea, as we go away from this thread of experience idea, we get something where we don't have the same kind of coherent existence. And it could very well be that it is sort of inevitable that that several things are inevitable. That it's inevitable from the Ruliad that there will develop self-reproduction, and that self-reproduction will lead to entities that have this kind of single threads of experience and so on. Once you have those single threads of experience, it becomes inevitable that entities like that will perceive a universe that has the laws of physics that we perceive our universe to have. But in a sense, the fact that we are inside that thread of experience and so on, that each one of us is inside such a thread of experience, that's a contingent fact. It's like saying, why are we living on this particular planet at this particular place in the universe rather than another one? That's something we can't have a theory that says that. We can say there could exist a planet that has these properties, but why we with our inner experience, get to be on this particular planet, that's not something about which one can have an external theory. And that's something for which it just happens that way. It's a contingent thing that just happens that way. So anyway, uh, that's kind of a few riffs, at least, on this question about the relationship between existence and the inevitability for existence, the inevitability of kind of this idea of threads of experience. This question, this point about the fact that once you have ideas about Rulial ensembles and so on, you inevitably develop this notion of being able to have something like this single thread of experience. There's much more to work out about this. This is a wonderful and very foundational philosophical kind of question. It's sort of almost theological. It almost rolls back the kinds of questions one's asking to the time before modern science in the 1600s to get back to questions that were asked for 1,000 years before that. So it's an interesting thing to try to explore. Let's see. Darth is asking, there must be a lot of embryo study in evolution. What are the driving forces when the concentrations of fluid to attract and assemble into complex organic molecules? Well, I mean, what I've talked about here really operates at the level of saying there is an overall process of self-reproduction running, and it's leading to adaptive evolution and so on. The underneath of that, of asking kind of how does that get started from individual molecules, we're working on that. I think there's some very interesting things that can be said. I don't yet know precisely what they are. Let's see. Okay, Fitz asking about multi-way systems, this idea of you can have multiple paths of behavior. Like if you're playing a game like tic-tac-toe, there are different moves you can make at each step, and you can build up this graph of all possible tic-tac-toe moves. So in a multi-way system, there's the notion of a branchial graph. We've talked about these extensively in connection with quantum mechanics, because in the multi-way graph that represents possible histories of the universe, the branchial graph is kind of a slice across that, and the branchial graph is talking about the relations between the different threads of history in the universe. And the idea is that quantum mechanics is kind of the story of how kind of minds like ours that span many, that sort of aggregate together paths in this region in this branchial graph, how minds like ours perceive this process of branching and merging that is summarized by the branchial graph. So the Fitz is asking, how does this branchial graph description how is the Rulial ensemble related to that? And how does that relate to the orchestration of entanglements in branchial space? That is a good question. And I think it's probably related to the way that observers like us sample quantum mechanics. And I think that's something that thinking about sort of the way that kind of patchiness develops in branchial space is probably something very related to sort of the emergence of objective reality in quantum mechanics, but I haven't untangled how all of that works. Hope to. Let's see. The LC is commenting, biology, life, evolution are going to push the computational bottom-up approach to the limits. I would think that things like coevolution, systems versus environment, levels of abstraction, and that this is a worthwhile pursuit. I agree it's a very worthwhile pursuit. I have to say that when one talks about biological evolution, I've mostly been talking about sort of one organism at a time evolution. There's the whole question about ecosystems, and then there's the whole question about sort of ecosystems in human society, the formation of human societies, the operation of economics, and so on. This is a thing that I'm hoping to tackle soon. And I think the same kinds of ideas are going to apply. the details of the simplest models, the minimal models will be different, but the goal is to be able to understand, just as the sort of key observation in biology that I've been addressing here is what is living matter like, and what is this gooey stuff that is the innards of biological organisms that is determined by large scale evolution. What's that like? So there are similar questions about economics, which which one can think about, and I've been trying to see how these ideas can apply there as well, and that's hopefully a coming attraction. All right. Well, thanks very much for joining me. And as I say, I realized as I was going through this piece of writing, this is a somewhat complicated subject, and I think this is really a very interesting kind of merging of a lot of different ideas, leading to some very interesting directions, particularly for biology, but also for thinking about sort of engineering systems that have been progressively refined and other kinds of systems that have some notion of adaptive evolution. Well, thanks for joining me, and bye for now.